Welcome. Today, we're diving into the world of biologic injectables, exploring how advanced analytical tools help us understand their character. I'm Dr. S.K. Singh, and together, we'll uncover the techniques that reveal the hidden details behind these complex medicines. Let's get started. Let's start by understanding what biologic injectables really are. These are therapeutic products made from living organisms, created through advanced biotech methods. We're talking about things like monoclonal antibodies, recombinant proteins, vaccines, fusion proteins, blood-derived products, and even gene therapy products. What makes them unique is their complexity. They have large, intricate structures that can vary and are sensitive to their environment. Because of this, analyzing biologics is much more challenging than small molecules, requiring detailed physical, biological, and immune system testing. When we talk about the global regulatory framework, we're looking at a set of key guidelines that shape how medicines are developed and approved worldwide. The International Council for Harmonization plays a central role here, alongside major agencies like the U.S. Food and Drug Administration and the European Medicines Agency. Their shared goal: to make sure every product clearly demonstrates its identity, purity, potency, safety, stability, and comparability. These criteria ensure that what reaches patients is both effective and reliable. When we talk about safety in biopharmaceuticals, several key attributes come into play. Endotoxins, sterility, and the risk of triggering an immune response are critical factors we monitor closely. Alongside safety, we look at critical quality attributes, things like molecular weight, the primary sequence of the protein, its higher-order structure, and glycosylation patterns. Purity is just as important, so we check for aggregates, fragments, and any leftover host cell proteins. Finally, functional attributes like biological activity, binding affinity, and overall potency ensure the product works as intended. Let's dive into molecular characterization using mass spectrometry. This technique lets us identify and analyze molecules by measuring their mass with incredible precision. It's like giving us a detailed fingerprint of the molecules in a sample, revealing their structure, composition, and even subtle differences. Mass spectrometry is a cornerstone in fields like chemistry, biology, and medicine because it helps us understand complex mixtures and discover new compounds. It's powerful, precise, and essential for modern molecular science. One key regulatory concern is that protein aggregates can increase immunogenicity, which means they might trigger unwanted immune responses. To monitor this, we analyze the protein purity by measuring the amounts of monomers, aggregates, and fragments. Using a technique called size exclusion chromatography, or SECHPLC, we can accurately quantify each component. For example, in this analysis, monomers make up the vast majority, around 98%, while aggregates and fragments are kept to a minimum. This helps ensure the safety and effectiveness of the protein product. Here's an example showing how we measure and control aggregates in a sample. The aggregate content is just 1%, well below the 2% limit we set. Because of that, the sample passes. We use several analytical techniques to get these results. SECHPLC quantifies aggregates. Dynamic light scattering measures particle size. Microflow imaging detects subvisible particles. And analytical ultracentrifugation profiles aggregation in detail. Each method gives us a different piece of the puzzle. Let's wrap up with an example that ties everything together. Here, the glycoform profile matches the reference standard, showing a clear distribution across G0F, G1F, and G2F types. This characterization is crucial because glycosylation impacts key drug attributes, potency, stability, how the body processes it, and immune response. To analyze these features, we rely on techniques like LC-MS, HPAEC-PAD, capillary electrophoresis, and HILIC-UPLC. Each method gives us a different lens on the molecule's complexity. Let's talk about why charge variant analysis matters. It's a key indicator of product consistency, helping us understand how much variation exists within a batch. Here, the main peak makes up 88%, while acidic and basic variants account for smaller portions. These differences come from chemical changes like demidation, oxidation, glycation, or modifications at the molecule's ends. To detect these, we use techniques like ion exchange chromatography and capillary isoelectric focusing. Together, they give us a clear picture of charge heterogeneity in the product. Now, let's talk about potency and bioactivity testing. These tests are crucial because they tell us how effective a product really is. Potency measures the strength, the amount of active ingredient that produces the desired effect. Bioactivity goes a step further, showing how the product interacts with biological systems. Together, they ensure what we're offering isn't just present, but truly works as intended. This step is key to delivering reliable and safe results. Let's talk about host cell protein analysis, a key part of ensuring product safety. These proteins come from the cells used during production, and they're considered impurities we need to control. We use techniques like ELISA and LC-MS to measure them accurately. For example, if we find 15 parts per million, and the limit is 100, we're well within the safe range. This analysis is critical because regulators focus heavily on these impurities to protect patients. Let's look at an example of stability indicating characterization. We start by measuring aggregates. Initially, they're at 0.5%. After applying stress, like heat, light, oxidation, agitation, or freeze-thaw cycles, aggregation rises to 2.5%. We evaluate key parameters, potency, aggregation, purity, and structural integrity. From this, we can identify the pathway that leads to aggregation, giving us insight into how the product changes under stress. This helps guide formulation and storage decisions. Let's talk about how we characterize subvisible particles, those tiny bits we can't see with the naked eye but matter a lot. We use several techniques: light obscuration, flow imaging microscopy, and resonant mass measurement. Here's an example from USB Chapter 788, where particles 10 microns and larger were measured at 15 particles per milliliter. The specification allows up to 25, so this product passes with room to spare. This testing is crucial because it helps ensure product safety and reduces the risk of immune reactions. When regulators come knocking, they zero in on a few key areas. They want to see solid analytical method validation, a thorough characterization package, clear comparability studies, reliable stability data, and of course, rock-solid data integrity. Common pitfalls: things like incomplete aggregate characterization, not enough glycosylation analysis, weak potency assay validation, and a lack of orthogonal methods. The trend now is leaning heavily on advanced analytical platforms. These tools help manage the product throughout its entire life cycle, keeping everything on track. We've drawn on guidance from leading organizations like the International Council for Harmonization, the FDA, and the World Health Organization to shape our approach. To sum up, biologic injectables are complex molecules that demand thorough analysis. Key quality attributes include identity, purity, potency, and stability, among others. Using advanced, complementary techniques gives us a full picture. Orthogonal methods are crucial. They ensure no detail is missed. Regulators expect a strong scientific rationale throughout development. Ultimately, robust characterization safeguards product quality, patient safety, and successful approval. Thank you for joining us today. If you need any further support or have questions, please don't hesitate to reach out. We're here to help. Thanks again for your time. It's been a pleasure sharing this with you.